First, it's a pleasure to be here. Oh, I blocked out one of the words there. Sorry. It's always a pleasure to come to the ISB and see the friends I have, and I want to thank Lee for the opportunity. A lot of what I think the introduction I was going to give, I don't need to give it because there's been, in the last talk, in a day full of tipping points, so I don't think it's so necessary. But what I'm going to talk about, the tipping points I'm going to talk about, are a little different from what we've been hearing about today. I'm going to talk about two changes that are associated with cancer. First is this process called chemically induced carcinogenesis, which has been a model system for understanding the transition from, say, cigarette smoking for healthy epithelial cells, in this case breast cells, to cancer. And then a second, which is associated with immunotherapy or targeted therapy, and is the resistant, the transfer from resistance to, from responsive to resistant. Okay. And so I'm going to talk about these in the context of, critical point transitions. And my background is as a physical scientist who came into biology. And Jeremy England gave this talk, and he gave an analogy like this, and I think I had better art, but it's just a great way to picture the stress between the two fields. So here we have a cat, and if you're a physicist, you might ask, can we approximate the cat as a spherical particle? What's the mass of the cat? What trajectory did it take off the building? Was it tossed or did it jump? And a biologist just asks, how's the cat doing? Which is actually a deeper question in the end. And so something that I want to try to do is to try to address questions and utilize data that has intrinsic value for both the biology but also from a physical perspective. Okay, so we're going to do kinetic studies, and we're going to do kinetic studies at the single cell level, and we're going to do those because you've heard about fluctuations today, but it turns out at the single cell level, if you measure the right things, one captures the fluctuations of the biology. And I'll go into why that's so important, but it turns out you can build up all of statistical mechanics from fluctuations if they're the right fluctuations, and that's basically the point. Okay, so we have these epithelial cells that we expose to this carcinogen, benzoazopyrene, and they evolve into this cancer phenotype after about 3 months by just observation by some colonogenic assay. The details of the process are that you expose them to this carcinogen for 48 hours, take away the carcinogen, let them go, and then 2 weeks later you do it again. 2 weeks later you do it again, and then you just let them go. So 3 rounds of toxin exposure, and then you let them go out for a little better than 3 months. And then we do this kinetic study with a control, and then looking out as we go out. And so so what we're looking here for, and we've also heard about correlations. And I think it's not always obvious why correlations are indicative of critical points, but they are. And I think an analogy that works for practically every single person that lives in Los Angeles, and I think most people that live in Seattle, is traffic. So if you go out on a freeway, when let's say it's a very early morning, 2 or 3 in the morning, there's no cars in the velocity of all the cars completely uncorrelated to each other. As you begin building up traffic, now these cars have local correlations in their velocities. As you continue to build up traffic, you get universal correlations, and then the traffic stops, velocity stops. That's the critical point. And so the characteristic of these correlations and a critical point is that they diverge as you approach the critical point. And that's the whole point of trying to look at correlations, because they have that predictive capacity. you can see something going to happen before it happens. Okay, so the way that we will look at these correlations in these cells, whether it's the drug resistance or the chemically induced carcinogenesis, is using a technology we developed in my group that now allows us to look at both metabolites and phosphoprotein signaling pathways from the same cells simultaneously. This is our most advanced form of the assay, which is about a 26-plex assay. We do on the order of between 1,000 and 10,000 cells in parallel. And the concept is pretty simple, and it's designed to allow us to measure these values in terms of copy numbers per cell. And we want that because we're trying to develop a physical model here. And you need absolute quantities. And so these things we're measuring, especially the phosphoproteins, are only present in maybe 1,000 copies per cell, which is very few. But if you put the cells into a little tiny volume, 1,000 copies is the detectable amount. And in that tiny little volume, we've patterned this 26-element antibody array that allows us to do either for metabolites competitive assays or for the proteins just simply sandwich ELISAs. And you can calibrate those, and so you can get copy numbers. Okay, so the way you get correlations is simply to plot one protein versus another and look and see how those two go, okay? And I'll make a point, especially later, that when you do bulk assays, you do not get correlations. You can see two proteins go up or go down, perhaps based on the stimulation, but at the single cell level, that does not necessarily reflect correlations, okay? And the correlations are particularly important here. Now, we're not looking at just two one-dimensional data of this guy versus this guy. We're actually looking at a whole matrix of data, okay? And so this is for chemically induced carcinogenesis. This is the panel of proteins we assayed, and this is the correlation matrix. And it turns out, from a measurement like this, one can get a mathematical description of the stable steady state. And the reason why you can get that is because when you look at the fluctuations, meaning, say, if I just look at phosphor-ERK, the abundance of phosphor-ERK versus frequency of observation, you should have a uniformly distributed set of fluctuations around an unchanging mean. If you have that, you have a steady state. And if you're measuring many elements of the cell, then you actually have a picture of the steady state of the cell, okay? And that allows you then to quantitatively assess the nature of perturbations. So as we send these cells through this process of chemically induced carcinogenesis, you could not ask for a more textbook example of a critical point transition. So you can see you've got a big time traffic jam coming, okay? And on top of that, if we go back a little bit, so I have this matrix that tells me that if I change the levels of phosphor-ERK, for example, I can calculate how the levels of these other proteins that are measured will change. So what that is telling me is the chemical potential of phosphor-ERK, okay? From a physical point of view, I have two quantities now. I have the abundance, the copy number, and the chemical potential. From the point of view of a physical system, that's an extensive and intensive property. It's like heat and temperature. Okay, so if we go forward, first you can see this traffic jam coming, but you can also see that you get particular sparseness at spots, and you see it right there, okay? That's actually the loss of a degree of freedom that's associated with the transition. And so if we look at the dominant modes from those correlation matrix, you can identify that you have what basically is the chemical potential of the proteins diverging, but the copy numbers are not really changing. And then you have a very, very significant drop. And so if you wanted to look at it as, say, a traditional biological signaling network, it goes from here to here to here to intensely correlated, and then you lose all correlations, okay? And so this analysis tells us that there's a critical point happening right in between this. I've just drawn this little a wave, a differentiated Gaussian here. We see a loss of degrees of freedom, and we also, at this point, actually see coexistence. And so one can analyze this exactly as a phase transition in chemical potential, okay? So what that means, if you think about ice water, you add heat to ice water, and the temperature doesn't change, okay? But if you add heat to water, the temperature changes. And you add heat to ice, the temperature changes. But at ice water, it doesn't change, and that's because you lose a degree of freedom, okay? So you can either change the copy numbers or change the chemical potential, and you don't change anything else in the matrix at this transition. Okay, so that's a model system, and it's a model system that at least biologists would agree is a definite cellular switch, that benzoazopyrene builds in a genetic instability into the DNA into these cells that causes this cancer to emerge. Okay, so I agree with Stephen Hawking. He says if you believe in science like I do, believe that certain laws are always obeyed. And then a good friend of Mike's, of Lee's and myself, Mike Phelps, this is not this guy. I don't know him. This guy, paraphrased. He said the basic physical chemical laws like quantum theory, thermodynamics are correct as far as we know. Biological systems exist in the physical world. Biology should follow the physical chemical laws theoretically. And then this guy, famous software guy, said, whenever anyone says theoretically, they really mean not really. Okay, so let's take these concepts and apply them to a more significant clinical problem. So I think many of you all are aware that immunotherapy, meaning harnessing the immune system to attack the cancer, has emerged and in many ways, for some cancers, changed the dialogue on cancer from a treatment to a cure over the past couple of years. But the cures are still the minority. So it turns out if you have a patient, and you really get patients with this level of metastatic disease, if they have an anti-tumor immune response that's baseline, that immunotherapy has a very good chance of accelerating that anti-tumor immune response and making those tumors dissolve. And that's about half the patients that come in for something like melanoma. Half of those patients appear to continue, and they have a durable response. They're effectively cured. But half of them will relapse. And if they relapse, they'll relapse typically in a year. And so we're going to talk about, and so then 25% of the patients relapse. We're going to talk about those patients. Okay, so it turns out that a very common form of resistance that's appearing in many of these patients has to do, and I think it's actually common in many cancers, has to do with the de-differentiation of the cancer cells upon the pressure of the drug. So this is an example of a patient before and after immunotherapy. And so this is an antigen that is reflective of a melanocyte, and in fact there are T-cell therapies based on this antigen. That's a de-differentiation marker. So it turns out skin cells, melanocytes, bear something in their lineage close to neurons, so you can go back to this neural crest lineage. And so in this case, there's not much neural crest, but there's a lot of melanocytes. After the influence of the drug, or the therapy, this was actually T-cell, anti-MART1 T-cell receptor therapy, now the melanocytes get really whittled down into some small area, and everything else now is expressing this neural crest marker. And you can see this in a big way here, okay? And so those cells that go through this melanocyte to neural crest transition now are basically hidden from the immune system, and they can go on to kill the patient. And it turns out that whether this just shows a commonality in melanoma. But so TNF has been implicated as the molecule from the immune system interaction that will promote this dedifferentiation. But it turns out many things do it, and this is verafenib, which is against BRAF mutant melanoma, and that also does the same thing. And so this is just a bunch of different cell lines. It turns out you can't really develop good mouse models, but you can certainly make good patient cell lines for melanoma. And they all show this same process of this dedifferentiation upon exposure to either TNF or verafenib. And you can follow it with flow cytometry using this melanocyte marker, which is on this axis, and this neural crest marker, which is on the y-axis. And so you more or less start up in this quadrant, which is melanocytes. And as you apply the drug, you just work your way around this loop. And so now these are neural crest ones that have no melanocytes. And eventually, you'll end up down here, where they're probably more dedifferentiated than the neural crest, but they are, this is the aggressive tumor, again, that you can't drug. So to get a grip on the kinetics of how this happens, the first thing we did was simply develop what's called a Markov model. And that says that, you know, if you have cells, wherever you place them on this map of melanocytes and neural crests, statistically, they can go one direction or the other. Their history doesn't matter. Only their future matters, OK? And so we purify these two populations and just watch them evolve for a few minutes, for a couple of days, and then we can develop these kinetic models. So this, it turns out you can see here, this is the differential drug response to these various quadrants. And so you get these models, of the kinetic process of the disease, and the models actually do a great job now. Just you go out like for 5 days and look at this, and then these models will let you predict out to many days past that, okay? And it's just a kinetic process. It just gives you the kinetics. And so with this kinetics now, we can define a time course for studying at the single cell level this melanocyte to neural crest transition. And so this is we take data points at all of these dots here. This shows basically how a resistant phenotype emerges. It emerges around something just after 2 weeks. And if you don't add the drug, then of course it's an aggressive grower from the beginning. And we're going to point out that and identify through the single cell analysis, the transition occurs way before. So if you'll go back, if you remember on this chemically induced carcinogenesis, that cancer phenotype emerges around day 90. You can see it in the collagenic assay. You can look at it and see, okay? You don't really see it before. But that transition that we see happens around day 9 or 10, okay? So the critical transition happens way before the phenotype emerges. And so this is a part of that kinetic series. And all I'm doing is plotting the correlations. And you have basically what looks like, again, a critical point. That's, again, almost a textbook example of a critical point transition. OK? Now, it turns out, if you look at the transition, or very, very close to the transition, you see this broad level of correlations, which is actually very noisy. But if you actually look as you approach the transition, you can identify signaling networks that are activated by the drug. Not repressed, activated. And those activated signaling networks are the machines that are carrying you through this transition. And so let me explain how that works. So this is what I meant by correlations and abundances. So here's an example of 2 proteins, or 4 proteins, A, B, C, and D, that are all at fairly high abundance. But these 2, if you look at single cell data, would be correlated. These 2 have no correlation at all. You can apply a drug, and all of these levels can go down. In fact, if they're phosphoproteins and the drug engages the target, the chance that all the phosphoproteins drop is pretty high. Okay? In fact, that's a good metric for drug engagement. But what you'll find is that some of these networks will actually become correlated. And that's the activation of the network by the drug. And some of them will lose the correlation, okay? And so seeing this kind of phenomena where you have an uncorrelated proteins that become correlated in the drug and eventually take you through resistance, this is actually not a rare phenomenon. We've been finding it in all kinds of cells that we look at, and all kinds of in tumor models as well, okay? And so when we analyze that melanocyte to neural crest, what we find is that the drug activates, in this sense, these two guys, okay? And it represses pretty much everything else, but it activates these two guys. And so we made a hypothesis that if we drug these two, as well as the initial drug, that we would have a good chance of stopping this melanocyte to neural crest transition and killing and continuing with tumor killing through the whole course of the treatment. And so this is the result of drugging individual or combinations. And so basically, you want to start and end at the same place. So here's before and during and after, but let's just look at before and after, okay? So you have dark, red, dark, and you end up with red, dark, red. That's bad. You want to be dark, red, dark, and you want to end up with dark, red, dark, okay? So this is the BRAF inhibitor. It actually seems to promote the switch. Trametinib, just against ERK, also dark, red, dark, red, dark, red, okay? You take this guy and this guy, well, that's really bad. You get really red, dark, red at the end, okay? But then you take all three, and you get dark, red, dark, and you end up with dark, red, dark, and you're able, that's this combination here, you're able to maintain tumor killing through the entire duration of the treatment. And it turns out it's not that unusual for this therapy to work, at least at the cell line level. And so here's these combinations of treatments. And at day 6, some of them have had differential effects. But by the time you get up to day 23, this combination of these three has actually been significantly better, except for this one patient sample. But it's been significantly better at repressing this transition, both in cholinergic assays and in these viability assays and cell killing assays that you see here. So see, you held up the sign, but I'm going to be done early.
Great.
Okay, so conclusions is that you can actually, the analysis of these protein-protein interactions, not protein levels, you can identify adaptive responses to drugs in a few days following treatment. And it turns out you should be able to. If you can identify when you have that critical point, and you identify when your correlations diverge, you go early on in that, and you identify those activated signaling networks. Okay? So it makes sense from you expect from the biological point of view that the cells are going to have a stress response to a drug to stay alive. You're more or less seeing that. But you can quantitate it in a more of a physical picture. Okay? And the statistical indicators and the biological indicators both point toward an upcoming transition. For chemically induced carcinogenesis and this melanocyte to neural crest transition, they really appear as almost textbook examples of a critical point transition when looked at this level of resolution. And then the detailed kinetic studies, the tumor cell responses to therapy, can yield guidance for combination therapies. And we've done this now for many patient samples from the operating room, as well as these melanoma models and GBM models. And it's a very, very robust technique. It doesn't mean you always come up with a good combination therapy to treat, but it certainly gives you a good shot. Like, for example, this particular drug, J, is probably not well tolerated. It's just a good experimental probe to use here. And so, we've now expanded this assay to a full 26-plex assay to try to get a better feeling for how to influence that more strongly. And it turns out that there are other similarities to these transitions that follow what one might expect of a physical system. So the chemically induced carcinogenesis is very irreversible. Basically, you're making a lot of mutations in the DNA. The cells become cancer. You You can't reverse that. But here's a kind of, this actually surprised us a lot. If you take the cells that you've differentiated all the way down to this very aggressive phenotype and release the drug, look, it just goes back. And it goes back in a way that has a different, if I did the Markov model here, it's a different Markov model than going forward. And so it acts as if there's a barrier with two steady states that are different energies. And so that barrier is faced differently by those two steady states. And so you get, but it's close. It's almost identical. OK. And so with that, I want to thank you for your attention. This is my group. And it turns out that the people that actually, so we live in a basement. We get to come out once a year, get our picture taken. Except for the people that actually did the experiments that I talked about. And they had to stay in the basement, I think. Natalie did some of the theory. And Yaping has been working on some of these, the melanocyte to neural crest. But everybody else was busy working that day and couldn't make it out, which it's sad because they're not in the picture, but it makes my heart warm as a PI to know that. And so with that, I thank you for your attention, and I'm happy to answer any questions.
Okay. Hi. I really like to talk. I was wondering, because earlier we were talking about networks and then those correlations. And these phosphoprotein networks are somewhat mapped out. If they could, if that's somehow worked in, or if there's a way of working that into the correlations. And also in that way, why you look at correlations of each possible pair, because only some you expect to correlate maybe?
So the phospho signaling networks are worked out to the point of molecules with sticks between them. Okay? And they vary highly from tumor to tumor. So if you go to those network diagrams, it gives you a pretty good guidance on the parts list to look for. But it does not give you guidance on what the structure of the signaling network will be. So the vast majority of these interactions are, I mean, you know, these networks act as excitable devices with excitability thresholds. And so they have, you know, nonlinear response functions. And so depending on where you are in terms of how hyperactivated that network is, it can look completely different than if it's sitting there at a stable, steady state that is, you know, not in a healthy cell. So I mean, because we get the network directly out of these measurements, because we get the, you know, you get it in one shot. And I would say it doesn't always look very similar to what you might expect from pulling down a signaling network off the web. It's not radically different, but, you know, the devil's in the details.
Yeah, I guess I was just wondering if there's some information in comparing them, or, yeah.
It's just the comparison is that the networks you would pull out of the literature give you a parts list, but they don't give you much more insight than that. If you know proteins form complexes, like mTOR, for example, forms big complexes, absolutely. You will always see those molecules be correlated. So that's a robust finding that will work its way through from the literature to these kind of results. Yeah?
Jim, why do you require so few measurements to say so much about networks? I mean, what's the logic?
So it turns out, if you look at the I have to draw it on the board. If you look at the fluctuations of a cell, of a particular protein, and you can make this many-dimensional by the number of proteins you measure. Okay. So here's copy number. This is, sorry, frequency of observation. And this is copy number. Okay. So you'll see, I can just make some histogram like this. Okay. So let's say this is phosphoprotein A. As I turn on the interactions of phosphoprotein A with other proteins, this guy will broaden, and it will broaden very rationally as I turn this on, okay? So one thing I'm able to do is by looking at these fluctuations, I have a measurement of this protein's interactions with every other protein it interacts with. I don't necessarily know those partners, but I know I've measured those interactions, okay? In addition, if I have a system that is responding to a perturbation, it's not at a stable state, okay, then you have a characteristic tail. You will always see this. Doesn't matter if it's cells, any physical system that's responding to the fluctuation will exhibit this tail. And so you actually have really, really deep insight. Now you take this one and you make it a 10, 12 dimensional, and our measurements have about an accuracy of about 5% in terms of copy number. It's actually a pretty good picture. I mean, I gave a talk, and Eric Lander said, well, why don't you just compare the genomes? I said, well, why would I do that many measurements when I can very quickly get that measurement out of a dozen proteins? And I don't know, I guess he likes genome. But he does. Yeah.
So I have a question about this protein correlation plot that you showed that shows the critical phenomenon. So basically, I mean, in general cases, there is a time lag between, there's a time between the information from one gene goes to the other one. So, for example, this one activates the other one, right?
Not for phosphoproteins. There is a time lag, but it's very fast. So we're not looking at protein expression. We're looking at post-translational modifications that happen within minutes of stimulation.
Okay, so it's because
That's
of
why.
the stimulation.
That's why these functional proteins and metabolites are such great indicators.
Okay.
If we were looking at expression of, say, housekeeping proteins that were just like membrane or something like that, you're right. You're right, it's too slow.
Yeah, I was assuming that you were telling that at the critical point that these time lags just go away because of the loss of the degrees of freedom, but that's not the thing.
No, that's not the case.
Okay.
You need to be, there's a very clear reason why we look at these proteins and not others, because they reflect the cell now, following the perturbation that just happened.
Okay, thanks.
I was wondering whether you find cooperation between your extreme cell types. So it seems like you have some reversibility, and I wonder like how predictable is this reversibility, and if maybe your extreme cell types, the melanocytes and the neural crest cells, are possibly cooperating, whether you can just inhibit this, So
that's a great question. So we actually have done experiments kind of like that, where we took, we were actually trying to explain tissue architectures. And with these platforms, we can do 2-cell experiments as well as 1-cell or 3-cell or whatever. And we can get lots of those. So we actually did like 100 experiments where the 2 cells were touching when they were 20 microns apart, 40, 60, 80, 100. And you could work out that when they were touching, they actually repressed their signaling activity, and when they were far apart, and it was gradual, they accelerated the signaling activity. You could fit those fluctuations to resolve the steady state. You could identify the steady state was when those cells were 100 microns apart. And you go to bulk tissue culture, and that is the characteristic separation distance. So we haven't done it yet for these phenotypes, but you absolutely can, through pairwise interactions, pull out steady state pictures that allow you to make predictions of what are otherwise seem to be complicated phenomena. And we've done that now for several cell lines, and it's a robust observation.
I mean, it's obvious that single-cell technologies have given this new granularity of how to study biology. We are doing stem cell differentiation as well. We can see very similar things for what you just described. My question is, many times, and we heard this before, there is a time between what we will measure in the molecular level and then the manifestation of the phenotype. And many times we have this collective phenomena in between in order to see the phenotype and the relapse you have in the end. So I was thinking, moving and thinking ideas for the clinic, what we miss and what we need to develop essentially to get to this time frame.
So I don't know. I mean, so as we've been looking at we've actually moved one of these platforms into a CLIA lab for analysis of GBM patients for combination therapies. And as part of the process of doing that, we've taken tissues from the operating room, looked at these phosphoprotein levels, say before and after therapy, and then looked at them in immunohistochemical staining of the tissues from the patient. We've been in agreement. So we haven't had and we've actually also done glucose and looked at FDG PET scans. And we've been in quantitative agreement as well. Now, one would argue that for these heterogeneous tumors that you might actually have to sample multiple regions of the tumor to capture that and be safe. And there's evidence for GBM, for example, that is so, that at least for 25% of the patients, you will get different answers in different parts of the tumor. I can't tell you how that's working out yet, but I mean, so far, these single cell measurements actually give you, and then the predictions we make, we test in vivo. Actually, I didn't do it for the melanocyte here, but we test in vivo for GBM, and they work.
Have you seen a personalized response?
Oh yes, for GBM, basically every patient we've looked at has been different. Doesn't mean there's always different drugs, but yes, absolutely.